Proteins can be categorized into two categories. They're either globular or fibrous, and globular proteins are compact and rounded and they're water soluble, and examples of these would be enzymes and antibodies. Or they're either fibrous, which means that they're elongated, insoluble in water, and they're physically tough, and examples of these would be collagen, keratin, and elastin. And amino acids are the building blocks of proteins, and they form together to create polypeptides, which are long chains of amino acids, which then become proteins. And the simplest amino acid is glycine. So you see that the variable R group of glycine is an H, but it can be many different things. For example, it could also be a methyl group or an ethyl group. And so amino acids combine together through condensation reactions. So in this example, you have two amino acids. So this is glycine and this is alanine. And so they come together. You see here, there's the carboxyl group, and then here's the amine group. And so in the condensation reaction, you lose one water molecule, so one OH and one H, which is water. And then the bond formed between the two amino acids is called a peptide bond. And so amino acids can either be polar, nonpolar, or electrically charged. And then in the cell membrane, you find nonpolar molecules within the membrane, nonpolar amino acids within the membrane, and you find polar amino acids on the outside of the membrane and also lining the transmembrane channel.
Different levels of the structures of proteins. So over here, there's this introduction. It says that protein structure can be described as 4 different levels: the primary, secondary, tertiary, and quaternary. So primary structure is just basically made of amino acids in a polypeptide chain, so it's like a straight line, and it's not really folded. It's just a very basic structure. But the secondary structure, you get either the alpha helix chain or the beta pleated sheet. So both of them use hydrogen bonds between the oxygen and hydrogen over here to maintain their structure and shape. But this is like a spirally chain shape, while this is like a sheet, and then there are like hydrogen bonds between each of the layers. And then for the tertiary structure, it is...
Hold on a second. I just want you to say one more thing. Like I want you to say that the secondary structure,
Yeah.
the bonding is from the carboxyl group on the backbone. You understand what that is?
Yeah.
I know you're not doing each other.
Yeah.
Like the backbone of the amino acid, not from the R group.
Oh,
Okay.
So the bondings are from the carboxyl group, but not from the R group. But in the tertiary structure, the bondings are between the R groups. So in the tertiary structure, the protein actually folds into a shape. It's just either globular or fibrous. So there are different types of bondings between different R groups. So there's the hydrogen bond between OH and O over here. There's the hydrophobic bond, which is over here. There's also the ionic bond, which is over here. And there's a disulfide bond, which is over here. So depending on the R groups, the protein would either be polar or nonpolar. And then over here, there's a quaternary structure. So this is a hemoglobin. This will fold into a hemoglobin molecule, and each of the groups here are called hemes, and these are tertiary structures. And that's the beta chain, that's the alpha chain, and then they will fold together. And then these are tertiary structures, so they'll fold together to become a quaternary structure, which is more complex than the tertiary structure.
So enzymes are globular proteins, and enzymes are basically just a biological catalyst. So catalysts, including enzymes, operate by reducing the activation energy of any reaction. And in the body, these reactions are very vital for life to occur, for like many metabolic processes, for example. So how do enzymes do that? How do they catalyze reactions? Well, there are 2 models to do this. There's the lock and key model and the induced fit model. So on the globular protein that is an enzyme, there's an active site. And at this active site, a specific substrate, which is basically what is going to be reacted, the reagent of the reaction, is bonded to the active site by either of these models. In the lock and key model, it operates rather like a lock and key, with the lock being the enzyme, the key being the substrate. In the induced fit model, it's slightly different. When the substrate approaches the enzyme changes its conformation of the active site to fit the substrate to help the reaction. Well, there are 2 ways that an enzyme can be inhibited. There's competitive inhibition and there's non-competitive inhibition. and they operate under different methods. So with competitive inhibition, the competitive inhibitor will actually bond the active site, but won't react to form any products. So because the competitive inhibitor is taking up the active catalytic site of the enzyme, the actual substrate with which the enzyme is meant to be, catalyzing, isn't being reacted in the enzyme. So the rate decreases. However, when the concentration of the substrate increases to a certain point, it just outcompetes the competitive inhibitor, so it can return to its maximum rate. With a non-competitive inhibitor, however, it binds to an allosteric site. An allosteric site is just another point on the enzyme, which changes the enzyme's conformation. And this can be a permanent thing. Effectively, it just basically destroys the enzyme's ability to act as a catalyst. So this actually effectively reduces the amount of effective enzymes inside a certain system. So it will actually reduce the total amount, the total maximum rate of reaction. There are also a couple of other factors that can affect the ability of an enzyme to function, and these are pH and temperature. At extremes of pH and temperature, the protein that enzymes consist of can actually denature, unfold, which again changes the active site. One example that is very important in the body, and it's an example of competitive inhibition, is end product inhibition. So this is when the end product of a metabolic process actually acts as a competitive inhibitor for one of the earlier steps in the process, so to prevent too much from building up. An example of this is ATP is a competitive inhibitor of phosphofructokinase, which is an enzyme in cellular respiration.